Ethel Browne Harvey was born in Baltimore, Maryland on December 14, 1885, the daughter of a physician, Dr. Bennett Browne, and his wife, Jeannie Nicholson Browne. Her early education was at the Bryn Mawr Preparatory School, a girls' school in Baltimore, Maryland. She then entered Goucher College and graduated in 1906. That fall, she enrolled as a graduate student in zoology at Columbia University and received a master's degree in 1907, followed by a PhD in 1913. At Columbia University, she first worked with Edmund Beecher Wilson and Thomas Hunt Morgan. She quickly developed firm research interests in their broad areas of cytology and embryology. While a graduate student, she published 6 papers, one jointly with Professor Morgan. Her doctoral dissertation, under the direction of Professor Wilson, was based on an extensive study of the cytology of the male germ cells of Nodonecta, a water insect. Browne Harvey first came to Woods Hole in the summer of 1906 to take a course being offered at the Marine Biological Laboratory. She then returned to MBL as an investigator every summer during her graduate work at Columbia University. Although in later years, she also worked at other marine stations, notably the Stazione Zoologica in Naples, she did return to MBL for at least a portion of every summer. In 1950, she became the first woman in a half-century to be elected to the Board of Trustees. Her only elected predecessor was Cornelia Clapp, who joined the board in 1900. Ethel Browne Harvey is well known for her experimental work on regeneration and development. She published her first paper in 1909, showing that a graft in the small invertebrate hydra could regenerate. She also conducted groundbreaking experiments on the embryology of the sea urchin Arbatia, mostly at MBL, and often traveling with fellow scientists to collect specimens. In 1927, she published her first paper dealing with the effects of the lack of oxygen on the development of Arbatia. Browne Harvey first used an optical centrifuge to layer and observe the cell parts of sea urchin eggs and remove the nuclei. She went on to show that the enucleated cells could cleave in the absence of the genetic material of the nucleus. She termed this method as parthenogenic merogamy, or division without fertilization, suggesting that other components besides the nucleus in the cell cytoplasm could control cleavage. I wonder what it would have been like to collect specimens during that time. Look at all those sea urchins. Oh, hi, Ethel. Have you found anything interesting today? Ethel was very enthusiastic to get her hands wet in research. She devoted several years to writing of a book entitled The American Arbatia and Other Sea Urchins. Dedicated to the memory of her former teacher, E.B. Wilson, the book deals not only with the experimental work on sea urchin development, to which she was so devoted, but also to the distribution, behavior, and general natural history of these organisms. Browne Harvey's lifelong dedication to science is clear in her personal choices as well. When she married fellow biologist E. Newton Harvey, their honeymoon was a trip to Japan to work at the Misaki Marine Biological Station, where MBL scientists Katsuma Dan and Shinya Inoue later spent time. During their research lives together, they shared laboratories at both MBL and Princeton. At MBL, Browne Harvey was one of the first to use an electron microscope, a revolutionary new tool for science. Ethel was elected to membership in the leading biological societies in this country and abroad. She was awarded as a fellow of the International Institute of Embryology and an honorary member of the Italian Society of Experimental Biology. And in 1956, Goucher College conferred upon her the honorary degree of Doctor of Science. In addition to being an extraordinary researcher and mother of 2 sons, Edmund and Richard, Browne Harvey was also an accomplished swimmer and tennis player. And for many years, Ethel and Newton presided over a corner table at the MBL mess. the old dining commons, where animated conversation accompanied every meal. Ethel Browne Harvey was an enthusiastic researcher who made significant contributions to experimental biology. Her detailed research with sea urchins at the MBL led to many publications in the Biological Bulletin over 20+ years. In a very real sense, Woods Hole was her scientific home. It was here that her most important work was accomplished, and it was here that she enjoyed her life in full measure. For these reasons, we remember and speak on the life of Ethel Browne Harvey at the Marine Biological Laboratory.